Hello students, in the previous videos I started with aerobic respiration. In that aerobic respiration we studied glycolysis, link reaction and Krebs cycle. Now in this video I am going to start with ETS, electron transport system and oxidative phosphorylation. ETS and oxidative phosphorylation. Okay, so first of all let's discuss why this ETS occurs. Actually, when the glucose undergoes breakdown through aerobic respiration, by the end of Krebs cycle it undergoes complete oxidation into carbon dioxide, water and energy. And that energy is released in the form of heat energy, which is trapped inside the cell in the form of ATP, NADH2 and FADH2. So this ATP is energy currency of the cell, whereas NADH2 and FADH2 are high energy intermediates. These are high energy intermediates. So ATP is energy currency of the cell. So why this is energy currency of the cell? Because when the cell is in need of energy to perform any other energy requiring process, immediately cell utilizes the energy which is stored in the form of ATP. But cell cannot utilize the energy which is stored in the form of NADH2 and FADH2. And hence this NADH2 and FADH2 undergoes breakdown through ETS, which ultimately results in the synthesis of ATP. So this is the reason why ETS occurs even after the complete breakdown of glucose by the end of Krebs cycle. Right. Now this NADH2 and FADH2 are called as high energy intermediates. Why they are called high energy intermediates? High energy because the amount of energy stored in NADH2 and FADH2 is more than the amount of energy stored in ATP. Because NADH2, one molecule of NADH2 is equals to 3 ATPs. And one molecule of FADH2 is equals to 2 ATPs. That means the amount of energy stored in NADH2 and FADH2 is more than the amount of energy stored in ATP. Right. So they are high energy intermediates. Intermediates because again they undergo breakdown or oxidation through ETS, which ultimately results in the synthesis of ATP. Hence they are called high energy intermediates. Right. Now what is ETS? ETS is a metabolic pathway. So this ETS is a metabolic pathway through which NADH2 and FADH2 are oxidized. Metabolic pathway through which NADH2 and FADH, NADH plus H plus and FADH2 undergoes oxidation, are oxidized and electrons are transferred from one carrier to the other carrier, transferred or passed from one carrier to the other and finally to molecular oxygen, resulting in the formation of metabolic water. To other and finally to molecular oxygen. Molecular oxygen is nothing but half O2, resulting in the formation of metabolic water. This half O2, after receiving electrons, it consumes protons from mitochondrial matrix and it is reduced into water. Right. So this is ETS. What is oxidative phosphorylation? So when electrons are transferred from one carrier to another carrier, when electron transfer occurs, protons are pumped into intermembrane space of the mitochondrion, which results in the development of proton motive force or proton gradient, which is utilized for the synthesis of ATP. So proton motive force or proton gradient is developed inside the intermembrane space of mitochondrion, due to which ATP synthesis occurs, which is utilized for the synthesis of ATP. So that is called as oxidative phosphorylation. So what is happening during this ETS and oxidative phosphorylation? NADH2 and FADH2 undergoes breakdown, oxidation through the complexes present in this ETS. And after breakdown, two protons and electrons and protons are released from this NADH2 or FADH2. So electrons are transferred from one carrier to the another carrier and finally onto molecular oxygen, which results in the formation of metabolic water. Whereas the protons are transported into intermembrane space. They are pumped by active process into intermembrane space of the mitochondrion, which results in the development of proton motive force or proton gradient, which is utilized for the synthesis of ATP. So now let's study the complexes which are involved, which are associated with this ETS. Okay, right. We know that this ETS occurs on the surface of inner mitochondrial membrane. Okay, so the site of this ETS is inner mitochondrial membrane. On this inner mitochondrial membrane, 5 complexes are associated with this inner mitochondrial membrane, which are involved in ETS and oxidative phosphorylation. Okay, let's see those complexes. So this is the complex, and these are electron carriers. Electron carriers present in this complex. Okay, so first complex is NADH dehydrogenase. So this NADH dehydrogenase is involved in the oxidation or breakdown of NADH plus H+, and the electron carriers present in this NADH dehydrogenase are FMN, that is flavin mononucleotide, and FES. Next, second complex. Second complex is FADH dehydrogenase or succinate dehydrogenase. That is FADH dehydrogenase, which is nothing but succinate dehydrogenase or succinate dehydrogenase. Okay, so this complex is involved in the oxidation of FADH2, and the electron carriers present in this complex are FAD and FES. These are the electron carriers present in this second complex, and the second complex is also seen in Krebs cycle, because in Krebs cycle also we studied about this succinate dehydrogenase. This enzyme succinate dehydrogenase is involved in oxidation of succinic acid into fumaric acid. So all the enzymes of Krebs cycle are present in mitochondrial matrix, but only one enzyme of Krebs cycle is attached to the inner mitochondrial membrane, and that enzyme is succinate dehydrogenase. So this succinate dehydrogenase or second complex is forming the link between both Krebs cycle and ETS, as it is involved in both Krebs cycle and ETS. So the second complex is forming the link. It is forming the link between both Krebs cycle and ETS. So this is about second complex. Now coming to third complex. Third complex is cytochrome C reductase. Which is also called as cytochrome BC1 complex. BC1 complex. Right. The electron carriers present in this third complex are cytochrome B, FES, and cytochrome C1. Next, coming to fourth complex, complex 4. Complex 4 is cytochrome C oxidase, which consists of cytochrome A and A3 net. So that is cytochrome C oxidase, which consists of cytochrome A and A3. Cytochrome A, A3 as electron carriers. Along with this A and A3, two copper centers are present. That is copper center 1 and copper center 2 are present in this fourth complex. So these are the four complexes which are involved in electron transfer. And there is one more complex, that is fifth complex, which is associated with this inner mitochondrial membrane. That complex is ATP synthase enzyme, which consists of two particles in it, that is F0 particle and F1 particle. F0 particle provides transmembrane channel for the transport of protons from intermembrane space towards matrix, whereas F1 particle is the headpiece which is protruded towards the matrix, and it consists of catalytic activity, catalytic site, which is required for the synthesis of ATP from ADP and inorganic phosphate. So totally 5 complexes are associated with inner mitochondrial membrane. Among 5 complexes, first 4 complexes are involved in electron transport, whereas 5th complex is involved in ATP synthesis. So now I will be giving you some more electron carriers along with these carriers which are associated with this ETS. So these electron carriers are mobile electron carriers. Mobile electron carriers. These mobile electron carriers are 2. They are ubiquinone. First carrier is ubiquinone, and second carrier is cytochrome C. So this cytochrome C is a small protein which is attached to the outer surface of the inner mitochondrial membrane. So these are the 2 mobile carriers which are associated with ETS, electron transport system of respiration, that is inside the mitochondrion. Whereas in case of photosynthesis, in case of photosynthetic ETS also, there are 2 mobile carriers, electron carriers. Those 2 mobile electron carriers are plastocyanin and plastoquinone. So this is about the carriers, electron carriers and complexes which are associated with ETS. So now I will be making a diagram to explain the mechanism of this electron transport and the mechanism of synthesis of ATP, that is oxidative phosphorylation, which occurs inside the mitochondrion. Okay, now let's study the mechanism of electron transport and the synthesis of ATP. So this is outer mitochondrial membrane, and this is inner mitochondrial membrane, and this is the space present between outer mitochondrial membrane and inner mitochondrial membrane. The space is called as intermembrane space. Okay, so here how many complexes are present, how many complexes are associated with this inner mitochondrial membrane means there are 5 complexes which are associated with inner mitochondrial membrane, and among these 5 complexes, first 4 complexes are involved in electron transfer, whereas 5th complex is involved in the synthesis of ATP. Okay, so this is the first complex, NADH dehydrogenase, second complex succinate dehydrogenase or FADH dehydrogenase, third complex, that is cytochrome C reductase, which is also called as cytochrome B7 complex, and fourth complex is cytochrome C oxidase, which consists of A1 A3. So cytochrome A1 A3 complex. Fifth complex, which is ATP synthase enzyme, which consists of ATP synthase enzyme, and it consists of 2 particles, 2 parts. They are F0 particle and F1 particle. F0 particle provides transmembrane channel for the transport of protons from intermembrane space towards F1 particle, that is towards matrix. F0 particle is embedded inside the intermembrane space, inner mitochondrial membrane, whereas F1 particle is protruded towards, this is called headpiece, and it is protruded towards matrix. Okay, this F1 particle consists of catalytic site which is required for the synthesis of ATP. Right, now let's study the breakdown or oxidation of NADH2 first and then FADH2. So let's study the oxidation of NADH2 And due to this, after this oxidation, how electrons are transported finally to molecular oxygen, transferred from one carrier to the another carrier, and finally to the molecular oxygen, and how protons are transported into, pumped into intermembrane space. Okay, now this is the NADH plus H plus, which is present in matrix, mitochondrial matrix, undergoes oxidation through first complex, resulting in the formation, in the release of NAD plus into matrix, and two protons and electrons are released into this complex. So two protons and electrons. Protons are pumped into intermembrane space. So here two protons are pumped. These are pumped into intermembrane space, whereas electrons are transported to the next complex through electron carriers. We already studied what are the electron carriers present in this first complex. They are FMN and FES. So electrons are transferred to FMN, then from FMN flavin mononucleotide. From FMN they are transferred to FES. From FES they are transferred to ubiquinone. So we already studied ubiquinone is the mobile electron carrier present in ETS. So when these two electrons are transferred, when they entered into ubiquinone, ubiquinone is actually a proton carrier. It is not an electron carrier. So when two electrons enter into ubiquinone, it consumes two protons from matrix. This is matrix. So from matrix, and pumps these two protons into matrix, but transfers electrons to the next carrier, next complex, that is third complex. So I just repeat. So when two electrons enter into ubiquinone, it consumes two protons from matrix, and it pumps those two protons into intermembrane space, but transfers electrons to the next complex, that is third complex. So what are the electron carriers present in third complex? They are cytochrome B, FES, cytochrome C1. I already gave you. So these electrons enter into first cytochrome B, then they enter into FES. After that they enter into ubiquinone. So when these electrons enter into ubiquinone, what happens again? This ubiquinone is mobile electron carrier. When they enter into ubiquinone, this ubiquinone consumes two protons from intermembrane space and pumps these two protons from matrix and pumps these two protons into intermembrane space, but transfers electrons to the next carrier of the third complex, that is cytochrome C1. From cytochrome C1, now these electrons are passed on to fourth complex via cytochrome C. Fourth complex via cytochrome C. So we already studied what is this cytochrome C. Cytochrome C is a mobile electron carrier, which is a small protein attached to the outer surface of the inner mitochondrial membrane. So this is the inner mitochondrial membrane. This is outer surface of inner mitochondrial membrane. This is inner surface of mitochondrial membrane. So now electrons are transferred from third complex to fourth complex via cytochrome C, which is a small protein attached to the outer surface of the inner mitochondrial membrane. So electrons, now in fourth complex, what are present electron carriers? Fourth complex consists of cytochrome A, A3, two copper centers, copper center 1 and copper center 2. So electrons enter into cytochrome A, then transferred to A3, then transferred to copper center 1 and copper center 2, and finally these electrons are finally transferred to molecular oxygen, that is half O2. So once this half O2 is receiving two electrons, once the electrons enter into half O2, it consumes two protons from matrix, and it is reduced into one water molecule. So now this water molecule, which is formed through this pathway, metabolic pathway, by the oxidation of NADH2 is called as metabolic water. So 1 NADH2 is equals to how many metabolic water molecule? 1 metabolic water molecule. So due to the oxidation of one molecule of NADH2, how many protons are pumped into intermembrane space? 6 protons are pumped into intermembrane space. So pumping is taking place. So the transport of protons from matrix to intermembrane space is by active process. So in the matrix, now the concentration of protons, the number of protons present in matrix will be reduced, is lesser than the number of protons present in intermembrane space, because there are two reasons. One reason is due to pumping of protons from matrix to intermembrane space during electron transport, and second reason is due to the consumption of protons by this oxygen to be reduced into water. Because of these two reasons, the number of protons present in matrix will be decreased than the number of protons present in intermembrane space. So what is happening? Proton gradient is developed inside the intermembrane space. That is nothing but proton motive force. So when proton motive force is developed, it is utilized. It is utilized for the synthesis of what? ATP. So this proton gradient undergoes breakdown. So after it undergoes breakdown, proton starts moving towards the fifth complex. Fifth complex. So fifth complex I already explained you. Fifth complex consists of two particles. They are F0 particle and F1 particle. F0 and F1 particle. So this F0 particle is providing transmembrane channel for the transport of protons from where? From intermembrane space to towards matrix. That is towards the F1 particle, which is facing towards matrix. So for every two protons that enter into F1 particle, so when the protons enter into F1 particle, this F1 particle, ATP catalytic activity of the ATP synthase present in this F1 particle gets activated, due to which ADP and IP together, inorganic phosphate together forms 1 ATP. So for every two protons that enter into this fifth complex results in the formation of 1 ATP. So how many protons are accumulated in intermembrane space? Totally 6 protons, which results in the formation of how many ATPs? 3 ATPs. So due to the oxidation of one molecule of NADH plus H plus, how many protons are accumulated in intermembrane space? 6 protons and 3 ATPs are synthesized. And the 2 electrons which are released from this NADH2 are finally transferred onto molecular oxygen. So after receiving these 2 electrons, molecular oxygen is consuming 2 protons from matrix and it is reduced into 1 water molecule. So this is all about the oxidation of NADH2. Now let's study the oxidation of FADH2. So FADH2 undergoes oxidation through which complex? Second complex. So FADH2 undergoes oxidation through second complex. So after oxidation, that is breakdown of FADH2, FAD plus is released into matrix, whereas electrons and protons enter into this second complex. This second complex cannot pump protons into intermembrane space because it is facing towards only matrix. It is not facing towards intermembrane space. Whereas in case of first complex, it is facing towards both intermembrane space and towards matrix also. Hence, it can pump the protons into intermembrane space. Whereas second complex cannot pump these protons into intermembrane space. So these protons and electrons enter into second complex from second complex. So what are the carriers of second complex? They are FAD and FES. So through FAD and FES, both of these protons and electrons are entering into third complex. In third complex, they enter into ubiquinone. So ubiquinone transports these protons into intermembrane space but transfers electrons to the next complex, that is fourth complex. As this ubiquinone is mobile electron carrier, it moves towards fourth complex also. So when it is moving towards fourth complex also, when the electrons enter into this ubiquinone of the fourth complex, what happens? It consumes 2 protons from matrix and pumps those 2 protons into intermembrane space but transfers electrons finally to the molecular oxygen, like this. In the same way, after receiving 2 electrons, this molecular oxygen is involved in consuming 2 protons from matrix, and this molecular oxygen is reduced into water in the same way. So 1 NADH2 is equals to 1 metabolic water and 3 ATP due to the pumping of 6 electrons into intermembrane space. due to the oxidation of one molecule of NADH2. Whereas due to the oxidation of one molecule of FADH2, how many protons are pumped into intermembrane space? 2 protons are pumped into intermembrane space. That is 4 protons totally. That is 2 protons from third complex and 2 protons from fourth complex. As 4 protons are pumped. So when one molecule of FADH2 undergoes oxidation through this ETS, what happens? 4 protons are pumped into intermembrane space, and these 4 protons results in the formation of what? Formation of 2 ATP. Because for the synthesis of 1 ATP, how many protons are required? 2 protons are required. So here also proton gradient is developed when the FADH2 undergoes oxidation. It is developed, proton motive force is developed in intermembrane space, due to which it undergoes breakdown, and these protons are again transported towards the matrix, that is into the F1 particle, through what? Through this transmembrane channel, that is F0 particle of fifth complex, and finally reaches F1 particle, that is headpiece of this fifth complex. So that one ADP and inorganic phosphate combine together, resulting in the formation of one molecule of ATP. So like this, this is about oxidation of NADH2 and oxidation of FADH2. Here I would like to give some points, like a quick recap. So here, transport of protons from matrix to intermembrane space is by active process, whereas movement of protons from intermembrane space towards matrix is by passive process. And whereas when one molecule of NADH2 undergoes oxidation, how many protons are transported into intermembrane space? 6 protons are transported into intermembrane space. So 4 protons are transported from first complex and 2 protons are transported from third complex. And when one FADH2 undergoes oxidation, 4 protons are transported into intermembrane space. From these 4 protons, 2 are transported through third complex and 2 are transported through fourth complex. So this is all about ETS and oxidative phosphorylation, that is synthesis of ATP. And I want to give you some more points related to this ETS. Now I would like to discuss some more points related to ETS. So actually what we are studying about aerobic respiration. So what is aerobic respiration? It is respiration which occurs in the presence of oxygen. But in this aerobic respiration, what is happening? The role of oxygen is limited to terminal step. So in this aerobic respiration, role of oxygen is limited to terminal step. Though the role of oxygen is limited to terminal step, the presence of this oxygen is vital. Terminal step, but the presence of this oxygen is vital because this oxygen drives the entire process of this aerobic respiration. How it drives the entire process of aerobic respiration? By the removal of protons or hydrogens from the matrix. So when the oxygen is removing or consuming the protons from the mitochondrial matrix, what happens? The concentration of protons present inside the mitochondrial matrix will be decreased than the concentration of protons present in intermembrane space of the mitochondrion. And hence, inside the intermembrane space of the mitochondrion, what happens? Proton gradient will be developed. Proton gradient or proton motive force is developed, which is utilized for the synthesis of ATP. So the presence of oxygen is vital because it drives the entire process of aerobic respiration, entire process by the removal of protons from matrix, mitochondrial matrix. So what can we say here? The final proton acceptor is oxygen, and even the final electron acceptor is also oxygen. Finally the electrons which are coming from the oxidation of NADH2 and FADH2 are reaching oxygen. So the final proton and electron acceptor, final proton and electron acceptor is oxygen. That is molecular oxygen, half O2. So this half O2, presence of this oxygen is vital for the process of aerobic respiration. Okay, now I want to give you a few differences between photophosphorylation and oxidative phosphorylation. So, photophosphorylation and oxidative phosphorylation. Okay, right. Photophosphorylation occurs in chloroplast. It occurs inside chloroplast, whereas oxidative phosphorylation occurs inside the mitochondrion. Okay, the proton motive force which is developed inside this chloroplast is developed by utilizing the light energy. So, light energy is utilized for the development of proton motive force in case of chloroplast. Proton motive force, that is nothing but proton gradient. And where this proton gradient or proton motive force is developed inside the chloroplast, it is developed inside the lumen of thylakoid membrane. Lumen of thylakoid membrane. Now coming to oxidative phosphorylation. So in case of oxidative phosphorylation, proton motive force is developed by the utilization of energy which is released from the oxidation of substrates. What are those substrates? Those substrates are respiratory substrates like carbohydrates, proteins or fats or organic acids. So here the energy which is released, from the oxidation of substrates, is utilized for the development of proton motive force or proton gradient. And where this proton motive force or gradient is developed? Inside the intermembrane space of mitochondrion. So this is all about aerobic respiration. So with this topic we studied glycolysis, link reaction, Krebs cycle, ETS and even oxidative phosphorylation also. Okay, in the next video I'll be starting with a balance sheet of this aerobic respiration. Hope you understood all these concepts. You go through all the videos one by one and grab the concepts which will be useful for solving the questions in very few seconds faster. Thank you, students.